Hello students, welcome back. We're going to start today with Signal Transduction Part 10. Signal Transduction Part 10. Let us do just a little review of the last chapter, of the last lecture rather. Okay. And so remember last time we were doing the alpha-1 adrenergic receptor. The alpha-1 adrenergic receptor, is it inhibitory or stimulatory? Alpha-1, it's stimulatory. So this is what happens with alpha-1. I'm just going to go through the steps here. Norepinephrine or some sort of alpha-1 adrenergic receptor agonist binds to the alpha-1 adrenergic receptor, turns the alpha adrenergic receptor on. When that receptor comes on, then GTP, then the G-alpha stimulatory, G-alpha Q. G-alpha Q comes to the receptor, then G-alpha GTP goes, activates phospholipase C. Phospholipase C cleaves a phospholipid in the membrane, PIP2, to create two things, DAG and IP3. IP3 goes, releases calcium from the endoplasmic reticulum. The calcium goes to calmodulin, activates calmodulin. Calmodulin activates CAM kinase. CAM kinase phosphorylates myosin light chain kinase. Then myosin light chain kinase phosphorylates the myosin light chain, and then we get contraction. And that's how the alpha adrenergic receptor, alpha-1 adrenergic receptor works. So I'm going to give you a little sentence here. It's not really a sentence, to help you remember that alpha-1 goes with Q. The beta-1 receptor, what G protein does it go with? Beta-1. What G protein does it go with? We only have two left, stimulatory and inhibitory. It goes with stimulatory. And what does stimulatory activate? What enzyme does stimulatory activate? What protein does it activate? AC, adenylyl cyclase, exactly. And so here, for alpha-1 here, the G protein is Q. So I always use this little sentence, this little phrase here to remember. Alpha-1. And you just have to remember it's not beta-1. So if you just remember this, 1, 2, 3, 1, Q, C. 1, 2, 3, 1, Q, C. So if you go back, what does all of that mean? So you know what 1, 2, 3 means, but 1, Q, C, alpha-1 simulates GQ, and GQ stimulates phospholipase C. So if you can remember that, you'll know that it's alpha-1, GQ, phospholipase C. If you remember 1, 2, 3, 1, QC. Kind of makes sense? So there you are. One thing I told you last time is that we know what IP3 does, and we said we weren't going to tell you what DAG does, diacylglycerol. But after thinking about it, that's real prevalent in biochemistry, so you kind of need to know what diacylglycerol activates. It activates a protein. Okay? And so I'm just going to put it out here somewhere. I'll put it right here. It activates a protein kinase. Okay, let's see. Let's see if you can remember this. Adenylyl cyclase makes more of what metabolite or what compound in the cell? Cyclic AMP. And what protein kinase does cyclic AMP activate? PKA, because it's cyclic AMP activates protein kinase A. Make sense? Now, we have DAG. DAG activates a protein kinase. You're going to get it wrong when I'm going to ask you, what is the letter of this protein kinase? You're going to get it wrong at the first time because you're going to be thinking a certain way. But then it's going to make sense to you, kind of. So, if you had to guess, what protein kinase, I need to see kind of more things. Okay. What protein kinase do you think is activated by DAG? What would make the most sense? Okay, what do you say? G. Why G? Okay, glycerol. And what did you say? D? C? Why C? Okay. Okay, because like phospholipase C has a C in it. Okay. Any other guesses? No one said the obvious one. D. Right. D because it's diacylglycerol. That's the one I expected you all to say first, but it's not D. It's not D. And then you said G because glycerol, that's not it. That's not it. But it is C. It is C because it's protein, it's phospholipase C ends up activating protein kinase C. So that is the enzyme, that is the kinase that's activated by diacylglycerol. And it goes on to phosphorylate lots of other things, activating lots of other things in the cell. Okay. The protein kinase that is activated by diacylglycerol is protein kinase C. Protein kinase C. Now you mentioned protein kinase G. In 2 days, in 2 lectures, we'll talk about protein kinase G and its involvement in the signal transduction pathway leading to penile erection, because that's kind of an important one in biochemistry because a lot of drugs are made there. And we'll talk about that later. Okay? Everybody okay with this? And so that's all I'm going to add for this one. And so now we are completely finished with alpha 1 adrenergic receptor. Question. Okay, so just it sounded just like a little rhyme to help you remember. The alpha 1 adrenergic receptor activates GQ. The G protein is Q, and then the GQ protein activates phospholipase C. So the sequence is alpha 1 GQ PLC. And so if you can remember 1, 2, 3, 1 QC. Yeah, they're Q here. Question. No. Protein kinases don't make cyclic AMP. Remember before, adenylyl cyclase makes cyclic AMP. The protein kinase C phosphorylates proteins, and it's going to phosphorylate proteins that activate this cell. But we just aren't going to talk about those because we already have a protein. We already have plenty of protein kinases. We need to know what they do. We have CAM kinase, and it phosphorylates myosin light chain kinase, and myosin light chain kinase phosphorylates myosin light chain, and then that causes contraction. Okay? Now, I don't want to say this, but protein kinase C could possibly phosphorylate calcium channels. And if it phosphorylates those calcium channels, just like in the heart, it opens them up, lets calcium in. If you have more calcium in, you're going to activate more calmodulin. If you activate more calmodulin, you're going to activate more CAM kinase. Then you're going to activate myosin light chain kinase. Then you're going to phosphorylate myosin light chain. Then you're going to get contraction. So all of these things, even though it's going by different fronts, they all lead to contraction of this smooth muscle cell. Kind of makes sense? And so that's the biochemistry of this signal transduction pathway. Now, let's go to, so those are all of the signal. We've done alpha 1. We've done alpha 2. We've done beta 1. Which one have we not done? Beta 2. And so there's also a beta 1 and a beta 3. But all the betas work by the same mechanism. They all go beta 1, G stimulatory. But you should be thinking in your mind, well, Dr. Merida, but beta 2 is inhibitory. But it just depends on how the cell is wired. If you stimulate that cell, then it's wired to actually turn things off. So we aren't going to talk about alpha. We aren't going to talk about beta 2 or beta 3. So those are all of the adrenergic receptor signal transduction pathways we need to know. Everybody okay with that? Now what we're going to do, we're going to do something slightly different now. We're going to take a kind of a big picture approach to things now. Okay. Question for you all. I'm going to draw something, and you all should be able to tell me what this something is. And then I'm going to ask you a question. Okay. Okay. What is that? What have I drawn? No, it's not acetylcholine. Maybe what if I drew it this way? I'm going to draw, it's this thing right here, but I'm going to draw it another way. Exactly. Okay. It's a nerve. It's basically a whole nerve. It's an entire nerve. But I've just drawn it this way because it's easier to draw that way. This is the body of the nerve, and this is the nerve ending. Everybody okay with that? Now I have a question for you. It's just a conceptual question to see where your thinking is. Now, this part of the nerve is the nerve ending, correct? What do nerve endings release? Neurotransmitters. Everybody okay with that? Okay, very good. Now, this is the question. Let's see where your thinking is. On the neurotransmitters, there are two options. The neurotransmitters can be synthesized here. They can be synthesized here in the nerve body, then transported down the axon to the nerve ending, and then released. That's one option. Or the neurotransmitters can be synthesized right in the nerve ending and then released. So those are the two options. Those are the two options. And I just want to see what you all think. Which option do you think? Only one of those takes place. So here are the two options again. The neurotransmitter can be synthesized in the nerve body and then transported to the nerve ending and then released. Or the neurotransmitter can be synthesized in the nerve ending itself and then released. Okay? Those are the two options. Does everybody understand the options? You're not getting a grade for it, so it's no pressure. I just want to see what you think. So, how many people believe that the neurotransmitter is synthesized in the nerve ending and then released? Okay, 1, 2, 3, 4, 5, 6. Okay? How many people believe that the neurotransmitter is synthesized in the nerve body, transported to the nerve ending and then released? 1, 2, 3, 4, 5, 6. So, you don't know the answer. No way you'll know the answer. I just want to see what you think. And so I think more people, I think there were 7 that said the nerve ending and 6 said the nerve, the soma, or the body. The answer actually is it's released. It's synthesized in the nerve ending and released at the nerve ending. That's where your neurotransmitters are synthesized. In the nerve ending and then released from the nerve ending. And so what we're going to do for this lecture is we're going to just look at a nerve ending. We're just going to look at a nerve ending and see what happens in a nerve ending. Okay? See how that neurotransmitter is synthesized. What neurotransmitter? Let's decide on a neurotransmitter we want to use. There are lots of neurotransmitters. Which one would be a good neurotransmitter for us to use? Norepinephrine. Why not use epinephrine? Exactly. Epinephrine is made in the adrenal gland. So we'll use norepinephrine. Everybody okay with that? Okay. So let's go to our next. Okay, so let's get rid of this. So now what I'm going to do, this is going to be my nerve body. That's going to be the body of the nerve. This is going to be my axon. Okay. Let me make that a little better. Okay, I'm going to make this real small here. This is my nerve body. And that's my axon. Okay, everybody okay with that? Now I'm going to draw my nerve ending. Is the nerve ending bigger than the body or smaller? Normally. It's smaller. But since I need to draw a lot in this nerve ending, I'm going to make it bigger. But you all are just going to, you all know, in your heart of hearts that it's not really bigger. Don't you? Excellent. Excellent. So this is going to be the nerve ending. So I'm making a really, really big nerve ending because I need to draw some things in it, not because it's bigger. Make sense? Excellent. Excellent. Now, how do we make... Okay, so if this is the nerve ending, then over here, let's say that this is the effector organ. That it has to associate, that it has to release those neurotransmitters to. Let's give it another color. This is going to be the effector organ here. And let's say that this effector organ, let's just say it's the heart. So, if this is the heart, what type of receptor is going to be found on this? What type of receptors does norepinephrine bind to on the heart? Which one? Beta 1, exactly. Now, I could draw it like this. If you all would, don't draw what I'm going to draw right now. Just don't draw this for right now. I could draw it like this the typical way. 1, 2, 3, 4, 5, 6, 7. And I can say, okay, this is a beta 1. Right? But that's going to get, because we're going to be drawing a lot of receptors, and that's going to get kind of cumbersome. So instead of drawing it this way, if I just drew this and said, okay, that's my receptor, would you understand that? Okay, so that's going to be our receptor. That's our beta 1 adrenergic receptor on the heart. So this could be any effector organ. It could be beta 1, it could be beta 2. It could be beta 2 on the smooth muscles, on the blood vessels going to our skeletal muscle. Or it could be alpha 1 on the blood vessels going to the stomach. But we're just going to make this simple and just do one of them. Beta 1 on the heart. Okay, now the first thing you need to do to make what neurotransmitter are we trying to make? Norepinephrine. Okay, now in order to make a norepinephrine, we have to start with an amino acid. It's made from one single amino acid. Does anybody know what that one amino acid is? Tyrosine. Exactly. And so what's going to happen, you're gonna go to, let's say, Hardee's or somewhere. You're going to eat a hamburger. You're going to chew it up. It's going to go into your stomach. You're going to digest it. It's going to go into your small intestines. You're going to cut it up into little pieces. And then you're going to take those tyrosine amino acids and put them in your bloodstream. And now those tyrosines in your bloodstream are going to go to where this neurotransmitter is. And there's going to be here tyrosine. And this tyrosine came from. So right now I'm not going to draw the structure, if that's okay with you all. I'm just going to draw TYR for tyrosine. Because we'll do the structures later. You all are already learning those somewhere else. So here we have tyrosine outside of the cell. Where did this tyrosine come from? Where do you get tyrosine from? Exactly. You eat it. It's an amino acid. It's protein. You eat protein. That's how you get your amino acids. Some amino acids your body can just make. Some of them are essential. You can't make them, so you have to. So here we have tyrosine. And there has to be a channel here that lets the tyrosine come into the cell. So here, let's just make ourselves a little channel here. Okay. So what's going to happen? This tyrosine is going to then come through this channel and into this nerve ending. What are we trying to make? What are we trying to make? Norepinephrine. So this is the first step. You have to get the tyrosine into the cell. Make sense? Okay. Now, the next thing that has to happen, tyrosine has an OH on it, but we're going to add another OH to tyrosine. And again, we're not going to really worry about the structures right now because I don't need you all. So there's going to be an enzyme here, and this enzyme is called tyrosine hydroxylase. Tyrosine hydroxylase. And it's going to add something to, oh, bless you, bless you. This tyrosine hydroxylase is going to add something to tyrosine. What is it going to add to tyrosine? If you just had to guess. You said a phosphate, I heard. What'd you say? An OH, what'd you say? OH, exactly. A hydroxyl, an OH. And so when you add a hydroxyl group or an hydroxyl group or a hydroxyl group, I don't really know which one it is, but it doesn't matter. When you add a hydroxyl group to tyrosine, you make a new compound, and that compound is called DOPA. Not dopamine, just DOPA. You're going to make a compound called DOPA. And so DOPA is going to be our first catecholamine. What's a catecholamine? We'll learn about that next lecture. Maybe. Well, I don't think we'll get too far on this one. And so tyrosine is going to be converted to DOPA using the enzyme tyrosine hydroxylase. What are we trying to make? Trying to make norepinephrine. Is DOPA and norepinephrine the same thing? No, they're not, because you can see they have different letters. And so we aren't there yet. We're not there yet. So the next thing that's going to have to happen, another enzyme is going to come in. Okay, another enzyme is going to come in. Let's get a good color here. And this enzyme is called DOPA decarboxylase. DOPA decarboxylase. DECARBOXYLASE. And so DOPA decarboxylase is going to remove something from DOPA. What? Exactly. It's going to remove a carboxyl group. Can that make any more sense? Exactly. DOPA is an amino acid. It is an amino acid because it has the NH. It has the NH3 group, and it still has. I wonder if it would help you. Even though I don't want you to know these structures, I'm going to just draw the structure as we go. As we go. So you can just see what's happening. So I'm going to just draw here NH. I'm just going to draw tyrosine. Oh, everyone wants to sneeze today. Bless you. CH. COOH. And then this is alanine. And then if we add a phenyl group here, we get phenylalanine. And then if we add an OH, we get tyrosine. And so that's tyrosine. Okay, just so you know what's happening. So the first thing that's going to happen, we're going to have tyrosine hydroxylase to make dopa. And so now the first thing that's going to happen is we're going to add an OH there. Now that's no longer tyrosine, that's dopa, but it's still an amino acid because it has the amino group and it has the alpha carboxyl group. Okay. Now the next thing that's going to happen, dopa decarboxylase. What is it going to remove? It's going to remove this carboxyl group. So now this carboxyl group goes away. And so this compound is now, we don't call it dopa. We don't just simply call it dopa now. What would be a good name for it? What would be a good name for this compound here? If you take the carboxyl group off of dopa, you make dopamine. Exactly. So now we have dopamine. And so this compound now is dopamine. Now, so you all will know here, I'm calling these catecholamines. If you have a benzene ring with 2 OHs on it, if you have just 1 OH on it, it's called phenol. But if you add 2 OHs, it's called catechol. It's called catechol. And so if you have something, so this is a catechol, and it has an amino group, so we call this a catecholamine. Lots of your neurotransmitters are called catecholamines. But we'll talk about that later. That's not something I need you to know. It's just an aside. Okay? Now what's going to happen within this cell, let's see if we can move this out of the way. What is that? There it is. Okay, I'm going to kind of move this out of the way for a second. Let's move this over here. Just for now. Just move it over. And we'll come back to this later. We'll do one more step today. So what's going to happen? Dopamine is a neurotransmitter that we can use. So now what we're going to do, this thing has become very important now. Now we want to store it in a vesicle. We need to store this new neurotransmitter in a vesicle. And so we're going to take dopamine, and we're going to put dopamine into a vessel. It's going to go through this little channel here. It's going to be really a transporter here. And now we're going to store it in a vesicle. And so what's going to happen, this dopamine is going to move into this vesicle. What are we trying to make? Norepinephrine. Have we made it? We don't have it yet. So we still have some ways to go before we get there. And so when we come back next time, we'll figure out how do you convert dopamine into norepinephrine. Does everything make sense? Wonderful, wonderful. I'm not going to ask you any questions today since you all had a quiz. You all have a great day.